Imagine this scenario. You've got a doctor, right? And they need to figure out some mysterious genetic disease really fast. For a patient who's critically ill, time is absolutely crucial.
Yeah.
Or picture scientists dealing with these enormous data sets, trying to map out tiny, tiny differences across, like, thousands of human genomes.
Right, to understand disease risk and things like that.
Exactly. It sounds almost like science fiction, doesn't it? But it's actually becoming reality.
It really is. And welcome to the Deep Dive, everyone. This is where we take complex source material, like the research we're discussing today, and really pull out the key insights for you.
Yep.
And our mission today, well, is to help you understand how artificial intelligence, AI, is totally transforming how we read our DNA.
Making genomics faster, more accurate.
And more equitable, too. That's a big part of it. You should definitely get some aha moments today.
I'm looking forward to it. So today, we are diving deep into the world of genomics. We'll be looking specifically at how identifying these subtle genetic changes, the process called variant calling, how crucial that is. For everything from diagnosing rare diseases quickly to, powering huge research projects. Right. And then we'll get into this groundbreaking tool called Deep Variant. It was developed by Google.
Yeah, it's a really interesting one.
We'll see how it's approached, this deep learning approach, combined with newer sequencing tech like long-read sequencing and this pangenome idea, how it's all changing the game.
Fundamentally changing the landscape, yeah.
So let's start with that basic concept first. Variant calling. What exactly is it? Our DNA code is, well, it's mostly the same between us, right?
Overwhelmingly similar, yeah.
Yeah.
But it's those tiny differences, the variants.
Yeah.
Sometimes just a single letter change in that huge long string of A's, C's, G's, and T's.
Just one letter.
Sometimes, yeah. And those are absolutely critical for understanding our health, our predispositions, why some people might get certain diseases. So variant calling is, it's the whole process of finding those specific differences. You compare an individual's DNA sequence against a reference genome.
And why is that so important?
Well, number one, diagnosing rare genetic diseases. These affect maybe around 8% of the population, so not that rare when you add them all up.
Okay, things like higher breast cancer risk or...
Exactly, or pulmonary arterial hypertension, conditions like that. Finding the specific variant can be absolutely life-changing for treatment, for management, and beyond just individuals. Variant calling lets researchers do these massive studies. They can look for genetic links to more common conditions, things that affect millions.
Okay, so identifying these tiny differences is clearly super important. How did Deep Variant manage to tackle this in such a new way? What made it a game changer?
Well, the really clever insight with Deep Variant, the core idea, was to frame this genetic puzzle as an image classification problem.
An image problem? How does that work with DNA?
Okay, so imagine you have all these short DNA sequences, the reads, that cover a specific spot in the genome. They pile up, right? Deep Variant basically turns that pileup of reads into a kind of image. Think of it like layered transparency stacked on top of each other.
Oh, like layers in Photoshop or something.
Exactly like that. Each layer, or channel as they call it, holds specific information.
Like what?
Okay, so one channel might show the actual DNA base, is it an A, C, G, or T?
Yeah.
Another channel shows the quality score, basically how confident the sequencing machine was about that base call.
Right, like is it a clear signal or kind of fuzzy?
Precisely. Then you have channels for mapping quality. How well did that piece of DNA line up with the reference? Which strand, like which direction was it read from? Does this specific read support the potential variant, or does it match the reference?
Wow, okay.
And even a channel just highlighting if the base is different from the reference at all. All these layers, these channels, combine to form this input image.
And then the AI looks at the image.
The deep neural network analyzes that image, yeah. Just like it might learn to identify cats or dogs in photos, it learns to see the subtle patterns that indicate a real genetic variant, distinguishing it from just, random sequencing errors.
That's a really different way of looking at it.
It really was. And it allowed Deep Variant to pick up on patterns that older methods they often missed.
And the results, when it first came out, they were pretty striking, weren't they?
Oh, absolutely. Right away, Deep Variant showed really significant accuracy improvements. There was this competition, the Precision FDA Truth Challenge back in 2016. Deep Variant outperformed the gold standard method at the time, GATK, on some benchmark data. And it won the highest performance award for certain types of variants called SNPs.
SNP is single nucleotide polymorphisms.
That's them, the single letter changes. And in one blinded test where they didn't know the right answer beforehand,
Yeah.
deep variant cut the total number of errors by more than half compared to the next best method. We're talking like 4,600 errors versus over 9,500.
Wow, that's a huge difference.
It's massive. But maybe even more important is the adaptability that comes with deep learning.
How so?
Well, you can add new custom channels to these images. So if a new sequencing technology comes along, that gives you a different kind of signal.
You can just add another layer to the image for the AI to look at.
Exactly. Or you can retrain the deep variant model on specific types of data. Say, data from a different brand of sequencing machine, or just looking at the protein coding regions, the exomes.
Ah, so it learns the specific quirks or error patterns of that data type.
Precisely. Which means it can maintain that high accuracy across lots of different situations. It's not just a one-trick pony.
That sounds incredibly powerful, almost like future-proofing the analysis.
Yeah, that's a good way to put it.
And that precision, that adaptability, it must have become even more critical when this next wave of sequencing tech arrived, the long reads.
Oh, definitely. Long-read sequencing, technologies like PacBio, HiFi, Oxford Nanopore,
Yeah.
they were another huge leap forward.
Because they read much longer pieces of DNA.
Much, much longer pieces compared to the traditional short reads. And that's their superpower, really.
Why is reading longer pieces so important?
Because there are parts of the genome that are incredibly complex and repetitive. Short reads just get lost in those areas. You can't figure out where they belong accurately.
Like trying to assemble a puzzle with only tiny blue sky pieces.
Exactly. Long reads can span right across those difficult regions. Think about things like segmental duplications or certain medically important genes that are notoriously hard to map, like PMS2.
PMS2, that's linked to some cancer risks.
Yeah, the one. And the difference is dramatic. There's a benchmark data set, Genome in a Bottle V4.2.1. It uses long reads.
Okay.
And just by using long reads, it expanded the coverage of the protein coding parts, the exons, by 16%. And for that tricky PMS2 gene, coverage jumped from about 26% with older methods to nearly 86%.
That's incredible, being able to see almost all of critical gene like that.
It's a massive improvement in what we can actually analyze reliably.
But there was a catch early on with long reads, wasn't there? Something about errors.
There was, yeah. Some of the early long-read technologies, while great at length, they sometimes had higher error rates, particularly for insertions or deletions, indels.
Indels, right.
So, for example, with early PacBio data, the chance that an indel call was actually correct, the positive predictive values, it was quite low.
Yeah.
Maybe around 1.4%, or in some analyses, maybe 22%, depending on the setup. Still not great.
So you get the benefit of reading long stretches, but you might introduce these specific kinds of errors.
That was the trade-off initially. But guess what came to the rescue?
Let me guess. Deep variant.
You got it.
Yeah.
This is where that adaptability we talked about really paid off. By retraining the deep variant models specifically on long-read data, like PacBio data.
It learned to handle those indel errors better.
Dramatically better. That positive predictive value for indels on PacBio whole genomes, it jumped from that low 20s percentage points up to over 97%.
97%, wow.
Yeah. So suddenly you could get all the advantages of long reads, accessing those complex regions, getting better coverage without the major downside of those unreliable indel calls.
That really unlocked the potential of long-read sequencing then.
Absolutely. It showed how deep learning could quickly adapt to and overcome the challenges of new sequencing technologies.
Okay, so we've got more accurate variant calling with deep variant, and we've got long reads letting us see more of the genome. What does this actually mean for patients? Can we see this impact in like a clinical setting?
Definitely. And there's some incredibly compelling examples. Think back to that doctor needing a fast diagnosis.
Yeah.
There was a case study involving a 13-year-old boy. He was suffering from decompensating heart failure, a really critical situation.
Okay.
Using Oxford Nanopore sequencing another long-read technology combined with deep variant and some other specialized callers.
They sequence his DNA.
They did. And they were able to identify a likely disease-causing variant in a gene called TNNT2. This gene is linked to dilated cardiomyopathy.
And how long did that take?
Get this. Less than 8 hours. From starting the sequencing to identifying the likely culprit variant.
Less than 8 hours. That's unbelievable.
It's incredibly fast. An ultra-rapid genetic diagnosis, they called it. And this was a collaboration, by the way, involving Stanford University and the UC Santa Cruz Genomics Institute.
Amazing. And that diagnosis actually helped the patient.
Directly. It influenced his care, allowed for timely, disease-specific treatment based on that genetic finding. It just perfectly illustrates the real-world, immediate impact these technologies can have when combined effectively.
That's incredibly powerful. Knowing the genetic cause so quickly must make a huge difference.
It can, absolutely.
But it also brings up another really important point. As we get better and better at reading individual genomes, we have to talk about the reference genome itself. Historically, these have been based mainly on people of European ancestry, right?
That's a critical issue, yes. For a long time, the standard human reference genome was largely built from a very small number of individuals, predominantly with European ancestry.
And what's the problem with that?
Well, the problem is that human genetic diversity is vast. If your reference doesn't capture that diversity, then your genomic analyses, your tools, they might not work equally well for everyone.
So genomic resources are like not equally powerful across individuals of all ancestries, as the research puts it.
Exactly. It creates a potential bias, meaning some populations might not get the full benefits of genomic medicine. It's a major health equity concern.
So what's the solution?
The solution that's gaining a lot of momentum is the pangenome.
Pangenome.
Yeah, the idea is instead of relying on a single linear reference sequence, we build a reference structure that incorporates genetic diversity from many different populations across the globe.
Like a more comprehensive, more representative map of human genetic variation.
Precisely. A reference that reflects humanity's diversity much better.
And I'm guessing long reads are important for building this pangenome too.
Absolutely essential. Especially technologies like PacBio Hi-Fi, which give you both length and high accuracy. Remember those complex repetitive regions we talked about?
The ones short reads struggle with.
Right. Those regions often contain a lot of the variation between different populations. Long reads are crucial for accurately mapping and assembling those regions to build a high-quality pangenome.
Makes sense. You need the long reads to bridge the gaps and see the complex differences.
Exactly. And to help with this, Google actually developed another tool called Deep Consensus.
Deep consensus.
Yeah.
Related to deep variant.
Sort of related in that it uses deep learning, but it's focused on improving the raw output from the PacBio sequencers themselves. It makes the initial long reads even more accurate.
So it cleans up the data before you even start assembling the genome?
Basically, yeah. It helps generate better quality genome assemblies, which is obviously critical for ambitious projects like the Human Pangenome Reference Consortium. They're building these high-quality pangenome references.
Okay, so better long reads feed into building better pangenomes. And how does deep variant fit into this pangenome picture?
Well, deep variant can then be used with these new pangenome references. Again, in collaboration with groups like the UCSC Genomics Institute, they've shown that combining deep variant with pangenome-aware analysis methods leads to even more accurate variant calling.
So using a more diverse reference helps deep variant do its job even better.
Right. It reduces the bias from using a single reference and improves accuracy, especially for individuals whose ancestry isn't well represented in the older reference genome. It's a key step towards making genomics more equitable.
That's really encouraging to hear. It seems like this deep learning approach that deep variant pioneered is not just a one-off tool, but more like a foundation.
It really is. It provides a robust framework that can be extended and adapted for new challenges in genomics.
Like what else is being built on this foundation?
Well, one really important area is cancer genomics. There's a tool called Deep Somatic.
Deep somatic. So for somatic variants.
Exactly. Somatic variants are the genetic changes that happen in cancer cells. Specifically, they're not inherited. Detecting them accurately is crucial for understanding how a tumor develops and for choosing the right treatments.
And deep somatic adapts the deep variant approach for that.
Yeah, it adapts the idea to compare the sequence reads from a tumor sample against reads from the patient's normal tissue. And it works with both short and long reads.
How does it compare to other methods for finding somatic variants?
It's shown really promising results, often demonstrating higher accuracy, especially for those tricky indels, compared to other established tools like Strelka2 or Claire's. So it's helping push the boundaries in cancer research and potentially diagnostics.
That's fantastic. Are there other applications beyond human health?
Oh, absolutely. This framework is incredibly versatile. Remember how deep variant can be retrained?
Yeah, for different instruments or data types.
Well, you can also retrain it for entirely different species.
Seriously, like animals or plants?
Yep. For example, researchers retrained deep variant using data from Anopheles gambiae. That's a species of mosquito that transmits malaria.
Okay. Why would you do that?
To get much more accurate variant calls in the mosquito genome, understanding genetic variation in mosquitoes is critical for efforts to control malaria, like tracking insecticide resistance.
And did it work? Did retraining help?
Dramatically. When they used the standard human-trained deep variant model on the mosquito data, the error rate, measured by something called the Mendelian violation rate, was over 15%.
Which is pretty high.
Yeah, lots of errors. But after retraining the model specifically on mosquito data, that error rate dropped to about 5.8%. And for high confidence variants, it went from over 5% error down to less than 1%.
Wow. So tailoring the AI model to the specific species makes a huge difference in
A
accuracy.
huge difference. It just highlights how adaptable this deep learning framework is. It's not just for humans. It has broad applications across biology, helping us understand the genetics of potentially any organism we can sequence.
That's incredible. So when we kind of zoom out and look at this whole story, deep variant, long reads, pangenomes, deep somatic, non-human applications, it's clear this has been a massive leap forward.
It really has.
And it wasn't just, a couple of people working in isolation.
No, absolutely not. It definitely required a dedicated, passionate team at Google, folks who were really committed to genomics and, importantly, to building these tools as open source.
Open source, so anyone can use them.
Exactly. Making them freely available was key. But honestly, the real game changer, the thing that turned these from interesting tech demos into tools having a real positive impact,
Yeah.
was the scientific community. Researchers around the world embracing these tools, adopting them, testing them, providing feedback, collaborating on extensions. That's what made the difference.
So it's that synergy between the developers and the users, the whole community working together.
Precisely. That collaboration was and is absolutely essential.
That's a great point to end on. So for you, our listener, especially if you're studying biology, just think about this. We are generating absolutely staggering amounts of biological data every single day.
More than ever before.
And as deep learning keeps evolving, as it gets integrated more tightly with these new sequencing technologies, what else is hiding in our DNA? What other signals might we be able to uncover next?
Yeah, or not just our DNA, but the DNA of, well, all life on Earth. What no biological mysteries are waiting to be unlocked now that we have these incredibly powerful AI tools to help us look?